So welcome to today's edition of our online seminar series, Emerging Topics in Biomolecular Magnetic Resonance. Just to remind you that we kind of follow the Gordon Research Conference policy, so no private audio or video recording of lectures by any means. And this is also not required because when speakers agree, we record the lecture and load it up to the ICMRBS YouTube channel. Also to remind you that we have a web page which is listed on top here called emergingmr.org, where you see the schedule and where you also find the links to the recorded lectures. So regarding question and answers, we use the Q&A. Alternatively, you can also raise your hand to speak. Today we have two fantastic speakers, Julie Foreman-Kay from University of Toronto and Galia Devoluchina from the University of California. And Julie will start today. So I'll stop this now. Let me quickly introduce Julie. Julie Foreman-Kay is program head and program head and senior scientist in the molecular medicine program at the Research Institute of the Hospital for Sick Children. She's also professor in the biochemistry department at the University of Toronto. And I just followed her over the years, and Julie has done fantastic work on dynamic proteins, in particular intrinsically disordered proteins, combining NMR, biophysical methods, and computational tools to really dissect these proteins and how they interact in dynamic complexes. Julie has also been awarded with a number of prizes. Just to mention that she's the recipient of the 2012 CSND Jean Monnery Fisher Memorial Lectureship and the 2013 Cellar Senior Scientists Award from the Cystic Fibrosis Canada. And she's also a member, I think, of the Royal Academy of Science of Canada. So it's a great pleasure and honor to have you, Julie. And please, we're interested to hear what you tell us.
Okay, thanks so much, Marcus, and thanks to all the other organizers of these emerging topics seminars. And I want to just start by saying that I'm going to be focusing on the NMR aspects of my work, and it's all in collaboration with Louis Kay, a lot of it with a joint postdoc of mine and his Tae-Hoon Kim, and I'll mention some of the other people as we go along. So I am having trouble actually moving the slide. There we go. Okay, intrinsically disordered proteins, or IDPs, and intrinsically disordered protein regions, IDRs, are thought to comprise about 30% of the human proteome, and they are associated with complex regulation that, when disrupted, can lead to disease. And NMR is really the only tool for atomic level insights into their fluctuating structures and their interactions due to their highly dynamic and heterogeneous conformations. So during the course of IDR function in biology, they undergo numerous transitions as a function of post-translational modification, binding of other proteins, changes of conditions, and these can be full or partial disorder to order transitions, in the case of partial, giving rise to dynamic or fuzzy complexes, even order to disorder transitions. But for the purpose of today, I want to focus on phase transitions, which move from a solvated mixed state to a demixed liquid phase separated state, where you have a solvent depleted, solute enriched phase and a solvent enriched, solute depleted phase. And these liquid phases can also become more viscous and gel-like and can also transition into fibers. So this biophysics of liquid-liquid phase separation for all kinds of biological macromolecules, proteins, nucleic acids, lipids, complex sugars, is involved in cellular organization and the formation of biomaterials. And in the case of proteins and protein nucleic acid phase separation, these are thought to underlie the formation of membraneless organelles, such as those in the nucleus, like the nucleolus, and speckles, paraspeckles, PML bodies, and in the cytoplasm, stress granules, RNA transport granules, germ granules, as well as phase-separated condensates comprising biomaterials such as the pore of the nuclear pore complex, the cytoskeleton, extracellular matrix. So these biological condensates are increasingly understood to be critical for biological organization. And in the case of RNA binding proteins, which are found in a lot of these membraneless organelles, we have folded RNA binding regions, we have a lot of intrinsically disordered regions. And so the phase separation of these proteins and RNA is driven by dynamic multivalent interactions of all sorts between the folded and disordered regions and nucleic acids. And these can involve charge-charge interactions, pi-pi, cation-pi, cross-beta, interactions of folded domains with short linear interaction motifs in disordered regions, and folded protein binding domains, or RNA, DNA binding domains for nucleic acids, or just nucleic acid base pairing. So in order to study these, we've chosen as our model system two RNA binding proteins, FMRP and Caprin-1, that have C-terminal disordered low complexity regions that we've predicted to phase separate using our predictor p-score, which is based on pi-pi interactions. And we have found that these C-terminal regions with a high prediction that are also disordered can phase separate in isolation and readily phase separate, forming these droplets in the presence of RNA. These two proteins we chose because they co-localize to stress granules, P bodies, and importantly for me, neuronal mRNA transport granules, and mutations in the low complexity regions of these two proteins are associated with autism spectrum disorder. And we have shown that there are regulatory effects of the phase separation of these regions on biochemistry that underlies neuronal function, translation, and deadenylation. And I may have time to get into a little bit of that, but I want to focus now on the NMR and the biophysics. And so if you put a solution of the FMRP low complexity region in an NMR tube at room temperature, and then you cool it, you can get phase separated droplets. And if you let that sit, you can get a bulk two phase separated system with the condensed phase ending up in the coil region of the NMR. So you can look at the condensed phase of these IDRs and compare them to the dilute phase. And here we have an M15 proton correlation spectrum. And you can see that the chemical shift dispersion in both cases is similarly narrow. We don't have any significant ordering upon phase separation. And this is consistent with the liquid nature of the liquid phase separated state and the rapidly exchanging weak multivalent interactions. And we can also look similarly at the Caprin C-terminal LCR in the black and the blue in the dilute and the green in the condensed phases, and you can see that there's global broadening, some residue-specific broadening, but the chemical shift changes are not significant, and it's still disordered. So we're interested actually in the interactions of FMRP and the Cavern-1 LCRs, and just by themselves, they don't interact as this ITC shows, but when you have multi-site phosphorylation on serines and threonines of the FMRP, you can get a very nice interaction, and you can also see that when you put them together with the multi-site phosphorylated FMRP LCR, they readily phase separate. And so we're interested in the molecular interactions governing this co-phase separation of FMRP and Cavern, and turn to NMR. So when we're thinking about NMR studies of IDR phase separation, you need a lot of protein. We're talking hundreds of mgs per mil. We have low complexity disordered sequences that give rise to fast amide hydrogen exchange rates, poor amide proton chemical shift dispersions, so poor spectral quality. So we could think maybe to slow the amide exchange with low temperature, but that increases the viscosity of an already highly viscous condensed phase, and we can measure the viscosity with insights from translational diffusion coefficients from pulse field gradient NMR, and you can see that the diffusion is close to 2 orders of magnitude slower in the condensed phase. We could lower the pH potentially, but phase separation can be pH dependent, particularly if it's phosphorylation dependent. And here we show that the Cavern and phospho FMRP co-phase separation is very pH dependent. So at nice NMR pHs, we don't actually see that co-phase separation. And this is because it's dependent on charge, and the pKa of the phosphate obviously is giving different charges at the lower pH. So what about carbon detect experiments? And so we turn to those. So a CON experiment, and here's a CON that gives us information on the carbonyl and amide nitrogen chemical shifts. And here, just in buffer, the Cavern 1 LCR spectrum, and you can see very nice resonances, and this has enabled the assignment of this roughly 100 residue protein, which is really a model for us because of its size for being able to study the interactions in phase separated states. And we don't see significant chemical shifts upon condensation, so we can transfer the chemical shifts. And so here we are looking at the N15C13 labeled Cavern LCR inside, partitioned into a condensed phase of the FMRP LCR. And here partitioned and co-phase separated actually with phosphorylated FMRP. So remember, FMRP doesn't bind Cavern, but it can partition into the phase separated, Cavern can partition into phase separated FMRP, whereas Cavern and phospho FMRP bind and co-phase separate. So we can compare those condensed states, and you can see that there's broadening in both, but the phospho FMRP context has more broadening. And we're going to interpret this broadening and reduced intensity as a measure of dynamic interactions. And unlike for folded proteins, where we think about using chemical shift perturbations as a primary tool to understand interactions, here we're using reduced intensity to get the interactions. And you know, there can be a lot of components of binding to a disordered region in terms of its effects on line shape, but primarily we're going to think about effects on the correlation time upon interactions. We can also have partial disorder to order transitions, chemical exchange between various states, and we can even lose intramolecular interactions and increase intensity, but there's a lot of complexity. But in general, we're going to be relying on intensity as a measure of interactions, because chemical shift changes are often very small for just dynamic interactions of disordered states. So here we can see that there are a lot of arginines, tyrosines, and phenylalanines that are significantly broadened in these condensed states relative to buffer. And to quantify, we have this intensity ratio where we divide the intensities from the condensed phase by the intensities in the buffer. And so if you look, we see that there are arginine-rich regions in the protein, in these yellow, and we have in these regions, we have the most significant intensity differences between the two FMRP and phosphorylated FMRP condensed phase, and that's likely sites of differential charge interactions. And then in the pink regions, we have these aromatic-rich tyrosine and phenylalanine-rich regions, where we have very similar intensity ratios, very broadened, showing that these interactions are really important for phase separation in general. So the arginines, tyrosines, and phenylalanines all have pi character. So this is reinforces this idea of the importance of pi interactions. And these aromatics that we see, we know that they're important because we can tyrosine phosphorylate them and change phase behavior. So what about testing arginine importance? We can make to lysines, and we don't then see interactions with ITC with phospho-FMRP, and it's lower by a FRET measure, but we can, and we also see that we don't see phase separation. So this is dependent on the pi character of the arginine guanidinium group. So we want to investigate that by NMR. And we can also induce phase separation of caprin on its own with salt. It's a very highly charged LCR, and you can see at high phosphate or high sodium chloride concentrations, we can get droplets in the wild type, not for the lysine mutations, but at low millimolar concentrations of pyrophosphate, we can actually induce phase separation. So we want to use NMR to look at these interactions of the pyrophosphate and with differences between lysine and arginine. And so as I showed before, NMR approaches avoiding amides, the CON, are valuable, but proton detect is more sensitive. And so Lewis, together with his postdoc, Leo Wang, and interacting with our joint postdoc, Tae-Hoon Kim, developed this proton detect version, the HACONHA experiment, which gives very nice spectra, and putting this MoCA inside a set of pulse sequences to give a set of tools for backbone assignment has enabled very easy reassignment of the caprin LCR resonances. Okay, so now we're going to use them. And what we want to do this time is use the dilute phase chemical shift perturbations as we add pyrophosphate to see if we can identify residues that are important. And so as we add pyrophosphate, what you can see is that there are some residues that have significant chemical shifts. And just looking as a function of residue, we see that residues in the N-terminal part of this LCR have the largest chemical shift perturbations, and there are 4 of the 15 arginines in this region. So we wanted to test our hypothesis that these chemical shift perturbations tell us something about the importance of different regions for phase separation. So we made different 4 arginine to lysine mutations for N-terminal, middle, and C-terminal regions, and this is our all 15. And then we monitored phase separation using optical density at 600, a turbidity assay. And what you can see for wild type is as you add sodium chloride, phase separation starts at about 500 millimolar with no with all of the arginines converted to lysines. We don't see phase separation out to beyond 2 molar, but the 4-arginine to 4-lysine mutant construct at the N-terminus is the most dramatic of the other 4R to 4K constructs, really confirming the importance of these interactions of the N-terminal residues that we were able to hypothesize via the NMR chemical shift perturbations in the dilute state. And the important point here is that we have sequence context being significant and not just composition. So I'm out of time, I think. And so I'm just going to summarize this NMR and the biophysics and say that the CON type experiments are really valuable for studying IDR phase separation. And we've used intensity ratios within partitioning or co-phase separating partner sample, identify interacting residues that are pi containing the arginine guanidinium and the tyrosine phenylalanine aromatic rings, and the 3D HACON-HA proton detected experiments that Lewis and his group have developed have significant sensitivity advantages over the carbon detect, and we can also use dilute phase experiments to look at the chemical shift perturbation to identify important residues, underscoring the sequence dependence of interactions driving phase separation. And so I'm just going to give a tease of some of the biochemistry results that we have on this system, showing that depending on the phosphorylation, serine threonine phosphorylation of FMRP, tyrosine phosphorylation of caprin, you can have different phase behavior, including subcompartmentalization that reciprocally regulates deadenylation and translation that are important for biological function via controlling local solvent environment and partitioning of components. And so you should have a look at these two publications, which is the work that I presented today. And I just want to acknowledge Tae-Hoon Kim, my joint postdoc with Lewis, Brian Sang, MD PhD student in my lab, Robert Vernon, and other members of my lab, as well as Alaji Ba, who helped us start on this project, who's now faculty at SUNY Upstate, and also acknowledge Leo and Ranjith and Jacob Brady from Lewis's lab, Geneviève Seabrook from Mitsu Ikura's lab, who got us started on carbon detect experiments, and Nachum Sonnenberg, who's been our translational collaborator, and funding sources. And thank you again for your attention.
Thank you, Julie, for such an exciting talk. So there are already quite a few questions besides what I would like to ask, I guess. But let's start from the beginning. So Walter Chassin asks, looking at the disordered regions of proteins in condensates is very cool. What happens when you run the experiments with the full-length protein?
Okay, so full-length FMRP is very poorly behaved, and caprin not as bad, but these are really large multi-domain with lots and lots of disorder. So we have not pursued NMR experiments on full length. We have done some phase separation experiments, you know, with some of those full length proteins, but we really have capitalized on the caprin low complexity region because of its importance with truncations and mutations being in that region highlighted for autism spectrum disorder association, and that region being necessary and sufficient for phase separation of the full-length protein. So we're using it as this 100 residue disorder region that we can easily assign and get interaction information.
The next question is from David Eliza. Thank you, Julie, very beautiful. How do you interpret the apparent difference in the ITC results? No interaction with unphosphorylated FMRP, with equivalent severe line broadening for phospho and no, it's gone, for phospho and unphosphorated FMRP.
Okay, so there's a difference between bimolecular binding and the ability to partition into a condensed phase. And I actually thank you, David, for that question, because it's actually was very interesting to us to see that Capron's LCR easily partitions into both non-phospho and phospho FMRP LCR, yet only binds in a bimolecular, in the dilute phase as a binding reaction to the phosphorylated. And so when you think about partitioning, it's really like using the other protein as a solvent. And so there are weak dynamic interactions, but not the same as a tighter, maybe more specific binding that you would get heat released in an ITC experiment. So I think it's these very weak solvent-like interactions that define co-miscibility and define the specificity for what proteins and what nucleic acids and what small molecules are going to be partitioning into the different biological condensates in our cells, which have hundreds of molecules. And recently we've seen work from Rick Young and his collaborators showing that drug molecules can selectively be enriched in these biological condensates. And so thinking about disordered protein regions as solvents, and also the other folded regions and the nucleic acids that make up the bulk of this solvating material, I think is going to be a really important way forward to understanding this specificity and also drug targeting.
Thank you. Then Haribabu Atranari asks, lovely talk. You mentioned that you use intensity as a marker for multivalent interactions. Since there is a decrease in intensity, would you expect to monitor is the interacting states using CEST-like experiments, or are there too many of these states to observe them? So CEST for phase separation and identifying the states.
So there are very, 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 very many states. So we're talking about disordered regions and high multivalency. We're talking about all of those different interactions that I mentioned: charge interactions, pi-pi, cation-pi, the hydrophobic hydrogen bonds, et cetera, and all of it is interacting very quickly. So I wouldn't think it would be that helpful. The other thing is that the dynamics, it's not clear that the interaction would be captured within the time frame that CEST or other kinds of NMR experiments on that range of experiments would be sensitive to. We have thought about looking for dispersions or other things, but it certainly not looking to be for this system a particularly valuable approach. But there may be other systems where it could be valuable.
Then Paul Robustelli is curious about PREs, essentially. So did you try to measure intermolecular PREs using sort of diluted with a non-labeled protein?
Yes, so we have measured PREs, and in particular, we're interested in seeing whether we can get some more specific kinds of interactions like NOEs or PREs to some of these pi containing residues, and we're currently analyzing some of that data now. But yes, I mean, PREs I think are a valuable tool for dynamic disordered protein interactions. They're hard to to understand all the components in terms of the quantification because of the complexity of the dynamics, but they do provide, I think, qualitative type of insights.
Thank you. Then Benjamin Lorton asks, have you examined the effect of methyl arginine PTMs within caprin or on its ability to phase separate?
So we haven't done that with caprin. We've definitely done it with FMRP, and arginine methylation significantly lowers the propensity of the FMRP LCR to phase separate. We're actually quite interested in understanding how those post-translational modifications might affect the pi interactions of the guanidinium group, and we're working with some groups to look at actually density functional theory in terms of guanidinium interactions. But I would, we also saw that with DDX4's low complexity and terminal low complexity region, that arginine methylation decreases the propensity to phase separate. So I think it's probably general. And I think other people have seen, like with FUS and other cases, that it decreases phase separation.
An anonymous attendee asks, thank you for the exciting presentation. I would like to know, how did you quantify the protein in the phase separated state?
Quantify meaning what the concentration is, or quantify?
It doesn't say, so I guess it's the concentration, yeah, because you had somewhere written 10 millimolar or something like that. Yeah.
Yes, so I mean, you know, there are a number of different ways that you can try and quantify, but getting like a standard curve, and then, you know, taking a small small amount of your sample and you know, doing ODs is one way. You know, we've actually tried an awful lot of ways to quantify protein concentration in order to do quantitative phase diagrams, and some of these methods I think are described in a paper, a collaborative paper from my group and Lewis's group with Jacob Brady. PNAS paper, so I would point you in that direction.
So there's still quite a few questions. So I think, Julie, you can look at them later. I would like to continue, but before that, actually, I would ask a question myself. So when you add RNA to the samples and do NMR, how bad do the spectra get? Or
it depends. You know, it depends what you're adding, and it depends you know, which protein you're using, but we can actually study in the presence of small RNAs, and we are moving in that direction to look at that. So it's not a total disaster. So yeah, so Marcus, I don't actually know technically how to save the chat questions, so maybe if you could and send them to me, I'd be happy to look at them.
We'll do that, yeah, sure.
Thanks everybody for the questions, and thanks for your time. I'm looking forward to Galia's talk. Okay.
Could I ask another one before we continue? And this is one that I always ask when it comes to phase separated systems. What are the concentrations you are using compared to the physiological concentrations, if they are known, of these systems? And a second question I had was, when you, in one of the examples, or maybe even both, I saw that normally one loses signals when one goes to the phase separated state, but it seemed that you had some signals which actually only were observable in the spectrum. I think it was just the proto-nitrogen HSQC spectrum when the phase separated state was formed. So that was kind of opposite compared to what it is normally that one sees. So
I'm not sure if it was a chemical shift change that made you think that something was coming up, but so far in our analysis, I don't think we have new peaks in the phase separated state. They may have some
Okay.
chemical shift changes.
Maybe I just look in the paper then.
Yeah, but the first question is, some of these RNA binding proteins that are found in cytoplasmic mRNA granules are actually very highly expressed in the cell. And so they can be at micromolar concentrations in the cell. And in the presence of RNA at low micromolar concentrations, we readily see phase separation of these RNA binding proteins. So the conditions are not so far off, right? And if you envision that there's an awful lot, there's an awful lot of concentrated everything in the cell that provides an additional force, I think, towards phase separation, plus lots of nucleic acids and lots of these other disordered region containing RNA binding proteins around that can come together. I think it's very easy to envision that physics can drive this inside a cell.
Okay, thanks.